Okay, this is Lecture 10, Sensory Physiology. So now that we have learned a bit about the electrical and chemical signaling in the nervous system, we're going to start to put it together for you in the basic pathways of the nervous system. And we're going to start with the sensory pathways, talk about some basics of sensory receptors, for actually sensing the information within and outside of the body. And then I'm going to give you some of the major sensory pathways and how these pathways are organized. We'll talk about somatic sensation or sensation of the body, and we'll very briefly introduce you to the special senses. So as a reminder, the nervous system is the master controlling and communication system of the body. It is specialized to quickly detect and respond to stimuli. So the sensory physiology is really the detection of the stimuli that the body needs to respond to. The response then will be the actual motor pathways or the motor output, the movement of the muscles, smooth muscle, cardiac muscle, skeletal muscle, in order for the body to respond to what it has sensed. So the nervous system is composed of the central nervous system, which is the brain and the spinal cord, and the peripheral nervous system, which are the peripheral nerves and receptors. Overall, when we look at where we are in terms of the overall picture of the nervous system, we're going to talk a lot today about the sensory stimuli through the afferent pathways, or the pathways that are entering the brain and the spinal cord. We will have a separate lecture then on the brain and the spinal cord, and a separate lecture on the efferent pathways or the motor pathways. So we look at a very simple pathway. We have a sensory receptor which is going to detect a stimulus. It will have an action potential which will activate the afferent neuron into the brain or spinal cord. An interneuron, which is within the brain and spinal cord, will do the integrating, and then the motor neuron will respond to activate the effector organ, muscles, or glands. So I will let you guys review this nervous system organization. I'm keeping it here in the PowerPoints just to continue to remind you how these sensory pathways go from the peripheral receptors and nerves into the spinal cord and brain, and then back out through the motor nerves to the peripheral effectors, organs, muscles, and glands. So we're going to talk about sensory processing today. Sensory processing is essential for regulating and coordinating the body's behavioral responses to the environment. Again, this is for maintaining homeostasis and generally understanding what is happening within and around our bodies. So some example functions of sensory processing include attention and arousal, so knowing what we should and should not pay attention to, knowing when we need to be in a more aroused or attentive state to respond more quickly, perceiving the world around us, remembering what we have been exposed to so we can respond appropriately the next time, and then the more complex type processing, which will link to our emotion and our behavioral pathways. So perception is the conscious interpretation of the external world created by patterns of activity in the brain. So we are interpreting the world based on the sensation that is coming in and creating patterns in the brain. It is limited, and perception is not perfect. So we are limited by the types of receptors that we have in the nervous system, what we can actually respond to. So for example, humans cannot sense magnetic fields or UV light. Other animals can, but humans, over the course of evolution, did not need to respond to magnetic fields or UV light, and therefore we don't have receptors for those. Doesn't mean that they are not there, just means that we don't perceive them. We're also limited by our level of sensitivity. So for example, we only hear a certain limited range of frequencies of sound. If anybody has dogs or other animals at home, you know that the human ear is not nearly as sensitive as the dog ear. So they can respond to a much wider range of frequencies of sound, and they hear a lot more than we do. So just because we don't hear it doesn't mean it's not there. And we are limited by our central processing and filtering. And this is where perception really does get interesting. So what grabs your attention, what you pay attention to, is based on your sensitivity, the receptor sensitivity that you have, and also your prior experience, your memories, your emotions, your attentive state at the time. And everybody will see something slightly different in the room. So there are some very interesting perceptual illusions. So for example, when you look at this illusion, do you see a white square? There is no white square here. There's only outlines created by the red image. So if I say the word Pac-Man, those of you who have played the Pac-Man game may now start to see these red dots, not as being covered up by a white square, but as Pac-Mans on the corners. So processing at the perception level is really about the sensation or the awareness of the stimuli and the interpretation of those stimuli. Here's another one for you. When you look at this blue and white image, do you see a vase or do you see two faces? So now that I've said faces, can you see the profile of the woman's face here? Or if I say vase and tell you just to focus on the white, you may just see the vase. And there can be a lot of interesting perceptions based on how your brain has decided to attend to, filter, and interpret the stimuli around you. So all of this at its very basic level is sensory processing, and it starts in the sensory neurons. Sensory neurons are cells located in the afferent division, or the sensory division coming into the brain and the spinal cord. The peripheral nervous system, then the afferent division of the peripheral nervous system, will receive information from sensory receptors. Sensory nerves then are bundles of sensory axons in the peripheral nervous system that will contact neurons in the spinal cord and brain. We have major divisions of sensory processing. We can sense things that are in our basic body senses. So somatic refers to body, and that is skin, muscles, joints, the general body sensations. We have sensation in our organs. Those would be visceral sensations. We generally are not aware of those sensations. Doesn't mean they're not there though. So we can sense things like blood pressure and the chemicals in our blood. We just are not aware of those sensations. And then finally, special senses, which we tend to focus a little bit more on intuitively, so it makes more sense to us when we talk about the vision or hearing or taste and smell. These are very vibrant sensations in the body, and those are through special sensory organs. So the organization of the peripheral nervous system is the afferent sensory division, carries sensory information from the receptors to the central nervous system. And I want to focus here on this word receptor. We've spent a lot of time so far in the nervous system talking about receptors at the protein level on cells. This is not that kind of receptor. So the receptors here we are talking about are special tiny cells or tiny receptors on cells that are going to be sensing stimuli. So these are not the, say, neurotransmitter receptors or the tiny channels that are within a cell. These are receptors in a different level. So we have to be careful to say sensory receptors here. So the somatic afferent nerves carries impulses from the skin, skeletal muscles, and joints. The special sense afferent nerves carry impulses from the eyes, ears, taste, smell. Visceral afferent nerves carry impulses from the organs within the body cavities, and they will not be consciously perceived. Sensory receptors are going to be located as special endings of a peripheral sensory neuron, or they may be a special tiny little organ all by themselves that are connected by junctions to the sensory neuron. So let's look at those possibilities. Oh, we've got that in a couple of slides. I'm going to show you them anyway, and then we'll back up. So the sensory receptor on the afferent neuron is here. There's the sensory receptor attached to the afferent neuron. The sensory receptor next to the afferent neuron will then be releasing neurotransmitter to depolarize or affect the afferent neuron. Okay, let's go back to where we were. So sensory receptors are responding to some change that is detectable, and they are going to convert that information from the environment into electrical signals. So the stimulus is the change. It's the change in pressure on the skin. It's the change in light on your eye. It's the change in sound within your ears. And it will be transduced or converted into electrical signals by the receptor. So receptors are very specialized, large structures. They're not the tiny proteins we've talked about on membranes. These sensory receptors are attached to or within afferent neurons, and they will produce depolarizing graded potentials or receptor potentials. They will then have sensory transduction, which will convert the stimulus, light, heat, sound, pressure, etc., into electrical energy. So receptor potentials are depolarizing graded potentials that activate sensory neurons. So remember that graded potentials are those small subthreshold changes in voltage. They don't have a refractory period, and they are subject to summation, just like graded potentials. They can be converted into action potentials within the sensory neuron if there are enough receptor potentials within a given time. If a receptor is part of an afferent neuron, then voltage-gated sodium channels will change local current flow. If the receptor is next to the afferent neuron, the neurotransmitter release will lead to current flow within the afferent neuron. So these we call trigger zones, and they will be on the dendrite regions, not the axon hillock like other neurons. So what we have then is a stimulus like mechanical pressure, light, heat, sound, etc., that will be converted by the sensory receptor into action potentials in the sensory neuron. So here's a stimulus, we turned it on, and then that created a graded potential or a receptor potential. A bigger stimulus will give a bigger graded potential or a bigger receptor potential. The graded potentials then, or receptor potentials, can then be converted if they pass threshold into action potentials in the sensory neuron. Smaller stimuli, smaller graded potentials will lead to a lower frequency of action potentials in the sensory neuron. Larger stimuli, for example, more pressure, more light, more sound, will be converted into larger receptor potentials and then converted into high frequency, very fast release of neurotransmitter, very fast action potentials in the afferent sensory neurons. If all of this sounds like Greek to you, you guys may want to go back and review your neurophysiology part 1 and part 2 lectures. So here's how it happens. We have a stimulus on this specialized ending of an afferent or sensory neuron. That stimulus will lead to a depolarization, which will be local, to that afferent neuron. If there's enough of it, then it will open up the voltage-gated sodium channels and get an action potential through the afferent neuron. If the receptor is its own little specialized tiny little organ, then it will release neurotransmitter as it's activated. That neurotransmitter then will be received by the sensory neuron and lead to action potentials in the sensory neuron. So we are at this portion of our pathway. We activated the sensory receptor with the stimulus, and we got an action potential through the sensory neuron. There are many, many types of sensory receptors. We could go on forever about these, and they're so fascinating if you look at their structure. Many, many different subclasses and different variations on sensory receptors across animals. This is really fascinating as well. Just a brief couple of examples. Photoreceptors respond to wavelengths of light. That's what we have in our eyes for vision. Mechanoreceptors respond to mechanical movement. For touch, for pressure, for muscle stretch, that makes sense to you guys, I'm sure. So when you push on the receptor, it will actually push open the ion channels, and that will cause depolarization of the cell. Sound is a little bit interesting. It also goes through mechanoreceptors, and what happens is that the sound waves travel through our ears, and they move these tiny little hair cells in our inner ears by fluid movement. So the sound waves cause movement of fluid, and then that creates the perception of sound in our ears. Thermoreceptors respond to heat and cold. Osmoreceptors respond to the concentration of solutes in our body fluids. Chemoreceptors respond to the presence of specific chemicals in fluids. So for example, your tongue and your nasal epithelium or olfactory epithelium have chemoreceptors for tasting and smelling. We also have pain receptors or nociceptors that alert us when something has been damaged in the body and we feel pain so that we can protect ourselves. So sensory receptors, the many, many different types, are each activated by a specific stimulus. So for example, photoreceptors are activated by light. They each have a different sensitivity, and this is called the adequate stimulus. So an adequate stimulus at a receptor will cause changes in electrical activity in the sensory neuron. Something that is not an adequate stimulus will be below the level of stimulation, and it will not activate the sensory neuron. Some receptors can respond weakly to other stimuli in addition to their adequate stimulus. So for example, if you close your eyes right now and you put just a little bit of pressure on your eye, you may be able to actually see light in response to that mechanical compression of the eye. That's coming from your eye, and so your brain perceives that as light. So photoreceptors will respond to light, but they also weakly respond to touch. Stimulus intensity is something important for the body to keep track of. So how strong is a stimulus? And the brain will keep track of this by how many receptors have been stimulated and by how frequent the action potentials are at those receptors. So the body also wants to keep track of, or the brain more specifically, wants to keep track of new information. So in general, we don't care much about old information, and there are many, many, many things that the brain decides to ignore if they are repeated over time. So there are two ways to respond to continuous stimuli. You can have receptor adaptation and neuron habituation. There is also neuron sensitivity, which is the opposite of habituation, and this is for certain stimuli an enhanced activation based on prior experience. So So let's do receptor adaptation first. So receptors can adapt to stimuli by decreasing the extent of depolarization or to sustained or repeated stimuli. So my example for this one is always, you're sitting in the lecture hall and somebody farts in the back of the room, right? So there's that initial smell that kind of, as the chemicals are permeating through the gases in the room, you'll get that smell in your nose, and it initially smells terrible. But over time, the smell goes away. Well, unless the room was really well ventilated and the doors were open, that smell didn't actually go away. What happened was your receptors are no longer responding to that smell the same degree as it did before. But if you walked out of the room and you came back in, you might then start to smell it again. So your brain is very smart about not responding to stimuli that are continuous. This particular phenomenon happens at the level of the receptor. So the receptor themselves no longer respond to the same degree as they did before. They just stop responding. There are two ways to do this. There are tonic receptors that adapt very slowly over time, so they tend to be used for more maintenance functions. For example, balance and posture information is always relayed to your central nervous system through stretch receptors. We don't want them to stop responding because we want to always be feeding back or sending information about balance and posture. But over time, you might start to respond less to these stimuli. Rapidly adapting stimuli will be through phasic receptors, and these will stop responding very quickly when a stimulus is repeated. So the clothing on your body, the background noise in the room, these are very rapidly adapting stimuli. So touch receptors on the skin, you won't feel the ring or the watch that you wear after you've been wearing it for a while, because your receptors on your skin will simply stop responding. And they do that very quickly when a stimulus is repeated. So phasic receptors are fast adapting, and they generally look like this. They will turn on when there's new information. If that stimulus continues, though, that's this green line here, they will stop responding. When the stimulus turns off, that's also new information. They might have a little blip of activation when the stimulus turns off. Some examples of fast adapting receptors are temperature receptors, olfactory or smell receptors, and touch receptors. Tonic receptors will respond to the stimulus and continue to respond the entire time that that stimulus is on, albeit they may slow down their response a little bit. This is for situations where continuous information is valuable. For example, in the example of proprioception, or where our bodies are in space, and stretch of muscles. Okay, same graphs, just a little bit more clear for you guys so you can review them later. Another way to stop responding to repeated stimuli is not through receptor adaptation, but by actually depressing synaptic activity. So this is neuron habituation. So repeated stimulus reduces activity through changes in the voltage-gated calcium channels in the presynaptic terminal. Now, if those sound Greek to you, go back and review your neurophysiology. The voltage-gated calcium channels are what cause the neurotransmitter release. So if we reduce the calcium channel activation, then we will reduce the amount of neurotransmitter release to a given stimulus. And that is called habituation. So we will habituate to continuous stimulus over time by decreasing synaptic activity as well. The last is the opposite of adaptation and habituation. So neurons can actually increase their responsiveness to something that is, by prior experience, deemed to be very important. So a very strong or a very irritating or noxious stimulus can actually prime the system to respond more the second time. Have you guys ever had this experience where you're kind of like going through the haunted house, right? So you kind of have this very strong stimulation and your body starts becoming sort of sensitized, and then anything that comes up will kind of make you jump. This is sensitization. So painful or strong stimuli will lead to serotonin release, that's a neurotransmitter, and then through a second messenger cascade, which happens to block potassium channels, this will prolong action potentials in the presynaptic neuron. So this is increasing the number of, increasing the amount of activity or increasing the length of action potentials in the presynaptic neuron such that we will respond more if we're primed by a painful or strong stimulation before. So adaptation, habituation, sensitization, these are all ways to change our response to stimuli. Let's talk now about the pathways of processing. So when we talk about processing sensory information, we can go through various complex or multi-level pathways. So the way that some textbooks like to organize this is by talking about different orders of sensory processing from the outside to the inside of the nervous system. So there's the first order sensory neuron, which will be in the periphery or outside of the spinal cord that will detect the stimulus. There's a second order sensory neuron that will be second in the chain that will be usually within the spinal cord or lower brain regions like the medulla oblongata. And they will be the intermediary before it reaches the higher orders of the brain. So then the third in the chain of processing will also be in the central nervous system and will go through, say, the thalamus, and this will continue, etc., until we get up to the cortex of the brain, where we will have perception and conscious awareness of the sensory stimulus. So the receptor level will be the first order. So that's out here. The example here is somatic sensation, so touch, proprioception, vibration, pain, temperature, coarse touch traveling into the spinal cord from the spinal nerves. This will be the first order going into the dorsal horn of the spinal cord. The next order will be within the nervous system, either the spinal cord or the medulla. We will have, that's the red neuron here. This one is going to cross over to the other side of the spinal cord or to the other side of the medulla before it travels up to the lower regions of the higher regions of the brain. Then those impulses will be transmitted to the higher regions of the brain, say from the thalamus and then up to the cortex. It's not until we get to the cortex of the brain that we have the actual conscious perception of the stimulus. What's interesting about these pathways is that the information going from the first to the second to the third, it's very carefully organized by receptor group and by location in the body. So as these neurons are traveling through the central nervous system pathways, they travel together on the same pathway, and they're organized into regions of the brain. So this creates maps of information in the central nervous system that is relayed through each location as the neurons are traveling through their first, second, third order locations. So the information on the location and the stimulus type is preserved as these pathways are traveling. We call this labeled lines, such that at any point in the pathway you could stimulate or ablate or remove some of these cells, and it would go to a specific location in the body. So you would lose, for example, information on what is in the tip of your finger throughout any region of this, any part of this pathway. This gets to the coding of a stimulus, which is how does the central nervous system know what the stimulus is and where it has been activated. So it's going to follow these pathways and follow the circuitry to know that. So it's going to depend on the pathway and the areas that it travels to. It will also relate to sensitivity based on how much of any one area of the brain is devoted to a particular region of the body or a particular sensation. So this, for example, is the somatosensory cortex, and mapped out here are regions of the somatosensory cortex that respond to touch sensation in various regions of the body. So if you were to stimulate here, you would have a sensation in your lower lip. Here, you would have sensation in your face. Here, sensation in your fingers. And regions that are more sensitive, for example, the face and the fingers, will have larger regions dedicated to them in the cortex than regions that are less sensitive. For example, the wrist is not as sensitive as the fingers. It has a smaller relative space than the finger region. So discrimination of a sensory stimulus, or being able to tell what it is, happens through something called receptive fields. So sensory neurons respond to stimulus information within small regions of input. So in any particular region of your skin, you will have a tiny area that is loaded with sensory receptors. The sensitivity of any given area is determined by the amount of receptors in that area. If you have more receptors in a small space, then that will lead to higher sensitivity. So touch is a very easy way to think about this. So if you think about the regions where you would really, really, really not want to get a tattoo, right? So everybody who has gotten tattoos knows that there are certain places that will be much more painful than other places, right? So think about getting a tattoo on your face or on your fingertips. These are super sensitive areas that would hurt a lot. But, forearm, upper arm, not so bad, right? So you have very sensitive areas on your face, lips, fingertips. These are areas that have many more receptors in that area, more density of receptors, but other areas that aren't so sensitive will have fewer receptors. So small, dense receptive fields will correspond to the most sensitive areas. For example, fingertips will have very small, dense receptive field areas. Large, diffuse receptive fields will correspond to the least sensitive areas. For example, your lower leg or your forearm. So here's a view of that. So here's fingertips, where if you look at any given space, you will have very small, dense receptive fields on your fingertips. What that means is that when you have smaller receptive fields, if you put two separate stimuli in that area of the skin, you will actually be able to perceive that there are two separate stimuli because you will have two separate neurons that are innervating that area. When you have less dense or larger receptive fields, you will only have one neuron innervating or surveilling that particular area. And given the same double stimulus, you will only have one neuron active. The central nervous system will only perceive one of those stimuli because there's only one neuron there to respond. So I also like to think of this as thinking about military troops that are assigned to survey a particular area. So if your sensory neurons are surveillance neurons, right? They're sent out to survey a particular area. And if you don't have a lot of troops, you have a small army, then you're going to send out one guy for a huge field, right? Well, when he's looking that way, he's going to miss stuff over there. He can only respond to a certain amount because he has too big of a geographical area. His receptive field size is very big, and there's only one guy in that receptive field that's responsible for that whole area. So he can make a lot of errors, and he's not going to be very sensitive, and he's not going to be able to respond to everything because he only has so much energy. But if you put two guys in that same big space. Well, one can look that way, one can look that way, and they can both now have responses within that area and send more information back to home base. So when you have multiple neurons, more density of receptors, more density of cells in a smaller space, then they will be more sensitive and can respond better than just putting one guy in the same amount of space. There's also something called lateral inhibition. So lateral inhibition helps to increase sensory discrimination when receptive fields are very dense. So the receptive field closest to the stimulus will be the most active, and the receptive fields nearby will be only weakly activated. So this is going to occur at the level of the receptors and the level of the central nervous system through inhibition. So here, for example, in a very dense receptive field, you've got 3 neurons that are all responding to this small area of the skin. Well, how do we keep from getting too confused and overactivating or overstimulating this region? Well, what happens is the neuron that is closest to the stimulus will send out inhibitory signals to the neurons nearby to decrease their activity, and then the brain will know there's a lot of activity from this one, just a little bit activity from these ones, and we can pinpoint the stimulus much better when lateral inhibition is there. As opposed to them all being active at the same level, there's a little bit of more fine-tuning of information when we have lateral inhibition. So there's a picture of the lateral inhibition, so they each inhibit each other, and the strongest transmission is what will be sent to the central nervous system. Okay, so let's talk about some major sensory pathways. So we're going to talk about somatic sensation and special senses. So somatic sensation is sensation that is in the body, skin, muscles, joints. This travels through somatic sensory neurons, which you may remember from your anatomy class. These are often called the pseudo-unipolar neurons, and they're going to be processed through the spinal cord and the brain. So somatic sensory receptors will be the touch receptors. So for example, we have hair receptors, which will respond to movement of the hair on the skin. Merkel's discs are touch receptors that respond to soft touch. Pacinian corpuscles, you guys may remember these from your skin anatomy, will respond to vibration and deep pressure. Ruffini endings will respond to deep pressure. Meissner's corpuscles will respond to soft touch or light tickles. These will be located in and throughout the regions of the skin. Their information from those receptors, from the touch receptors, will be relayed through pseudo-unipolar neurons. These neurons are myelinated, and they have a very large cell body that is located in the dorsal root ganglion next to the spinal cord. Their action potential will be initiated at the peripheral end of the neuron near the receptor over here. They will then send the action potential through and into the spinal cord. So here I need to remind you guys about white matter and gray matter. So white matter in the central nervous system are called tracts. These are axons. White matter in the peripheral nervous system are nerves. Gray matter in the central nervous system are called nuclei, and don't get that confused with the nucleus of a cell. This will be groups of cell bodies in the central nervous system, and they will look gray or darker in color. Ganglia will be groups of cell bodies in the peripheral nervous system. So when we say dorsal root and dorsal root ganglia, the dorsal root is the axon containing the axons of the sensory afferent neurons coming into the spinal cord. Within the dorsal root ganglion, you will have all of those large cell bodies of the pseudounipolar neurons. So the sensory information is going to come in through the dorsal root ganglion, through the dorsal root, and into the dorsal spinal cord. The ventral root of the spinal cord, on the other hand, is going to be motor axons coming out of the spinal cord, and more on these later. So the gray matter of the spinal cord maintains organization of body region and type of stimulus or type of motor activity. So this back region of the spinal cord is going to have sensory information, or the dorsal horn of the spinal cord will be sensory, and the ventral horn of the spinal cord will be motor. The lateral horn of the spinal cord will also be motor, but it's going to be autonomic, or motor going out to the organs in particular regions of the spinal cord. So even at the level of the spinal cord, we start to have special maps that will contain information and segregate information as it is coming in from the periphery and going out of the central nervous system. There are also white columns in the spinal cord, and these are going to be ascending tracts or groups of axons that are traveling up the spinal cord. So think of this like an elevator, right? The information is going to be going up and down the white matter of the spinal cord. There are three main tracts or pathways of the spinal cord that you may hear about. The posterior white columns, or the columns in the back of the spinal cord, will have information on fine touch, pressure, and vibration going up to the brain. The spinothalamic tract will have information on pain, temperature, touch, and pressure, and I'll show you a picture of that in just a second. And the spinocerebellar tract is going to have proprioception, or information on the position of muscles, tendons, and joints coming into the central nervous system and going up to the brain. So first, the posterior white columns. These are the first ones we talked about. So we'll have the sensory information of touch, vibration, pressure coming into the dorsal root of the spinal cord, and then going into the posterior white columns and traveling up to the lower regions and then the higher regions of the brain. So here we have a first order, a second order, and a third order sensory neuron, which ultimately is going to carry information up to these mapped regions of the brain about this somatic sensation of these different regions of the body. The lateral spinothalamic tract will be the pain and temperature sensations from one side of the body going into the dorsal horn. So there's your first order sensory neuron. And then they're going to cross over and go up the lateral white columns, travel up the lateral white columns, and then to the second, through the second order, and then the third order sensory neurons up to the brain. Then we have spinocerebellar tracts, which are going to be giving proprioceptive, or information on where the body is in space, information on stretch of muscles, position of the joints. That's going to come in through the dorsal horn as well, and it's going to go up the spinocerebellar tract to the cerebellum, where the cerebellum will be coordinating balance and posture movements. So those were all ascending tracts, which are going to be sensory tracts. After that, the nervous system will integrate and decide what to do, and then send information down the descending tracts, which include the anterior corticospinal tract and the lateral corticospinal tract. We'll talk a bit more about these when we get to the efferent nervous system. So as a reminder, the spinal nerves. Spinal nerves are mixed sensory and motor axons that are coming to and from the spinal cord. We number them. And they are somatic sensations to and from the skin, muscles, and joints. They are also containing in certain regions of the body autonomic axons, which are going to be autonomic control of the organs. Cranial nerves, those are the ones you guys numbered in your anatomy class, cranial nerves 1 through 12, are going to be nerves to and from the brain without using the spinal cord as an intermediate. And these are going to be going to and from regions of the head and neck, including the special sensory organs, the head and neck muscles, head and neck sensory control, and autonomic control of certain organs. So from the somatic sensation and the basic pathways, I'm going to spend the next few moments, we're going to fly through the special senses. I wish we had more time for special senses, guys, but this is better than nothing, okay? So the special senses are located in special sense organs, and they're going to bring in sensory information from the environment to the central nervous system. So they are olfactory sensation, gustatory sensation, auditory sensation, vestibular sensation, and visual sensation. So the olfactory system is bringing in smell information, which is actually chemicals that are dissolved in the olfactory mucosa or in the cells of the nose. So these chemicals will be dissolved and they will be sensed by special sensory receptors, the olfactory receptors in the nasal epithelium. The olfactory epithelium then will transmit this information in through cranial nerve 1, which is actually a group of olfactory nerves, and up to the olfactory bulb of the central nervous system. The olfactory bulb then will take the olfactory information and pass it into the brain to the olfactory cortex, where we will become aware of that smell. Hopefully it's a rosy smell. Then we have the gustatory system. So the special sense of taste is in the gustatory system, and here it's food, but it's actually the chemicals in the food that are dissolved in the saliva of the tongue. So we have special receptors in the taste buds of the tongue, which are gustatory receptors, and they will send information through cranial nerve 7, 9, and 10 up to the medulla. From the medulla, that information will go up to the thalamus and then to the gustatory cortex, where we'll be aware of the taste of the food that we are eating. The auditory system is the special sensation of hearing, and this is sound. And it turns out that sound is actually transmitted through fluid vibration in the inner ear. There's a special organ within the inner ear called the cochlea, which as the sound waves are transmitted through the inner ear, it vibrates fluid within the cochlea. And there are tiny little cells called hair cells. They don't actually have hair on them, but they have mechanical receptors on them that will move when the sound vibrates the fluid. That information will be transmitted from the cochlea through cranial nerve 8 into the medullary olives, the inferior colliculus, and then the thalamus of the brain, up to the auditory cortex, where we will then be aware of the sound. The vestibular system is a special sense of balance and equilibrium. So what the vestibular system is sensing is the location of your head in space. If any of you get seasick, you can blame your vestibular system. So as your head moves, this also moves fluid in the inner ear, and that activates a different set of hair cells within the inner ear in a set of organs called the semicircular canals. And notice these are in the X, Y, and Z planes, so they can know when your head is moving in the X, Y, and Z planes of direction. Those will be transmitted through cranial nerve 8 as well into various regions of the brain, through the pons, the medulla, to the cerebellum, and also to the cerebral cortex. The last is the visual system, and this is our special sense of sight. So the special sense of sight is through the stimulus of light through the eyes. This activates photoreceptors in the eye, and that will activate optic nerve or cranial nerve 2, which will go through the thalamus and then to the visual cortex. Then we'll be aware of the stimuli in the visual cortex. Light information also goes through the brainstem to the pineal gland and to other regions of the brain to help the brain regulate day and night cycles. Here's a visual of the visual pathway. You can see the light in different regions of the eye will enter through the eye, travel through optic nerve or cranial nerve number 2. Some will cross over, some will stay on the same side, go through the thalamus, and then all the way to the back of the brain in the visual cortex. All of this sensation will be used by the brain in different pathways. So the simplest pathways are reflexes, and they are made up of the fewest cells. So from stimulus to receptor to the sensory cell is usually just a single cell in a reflex. That will then go to a single central nervous system cell, which will then immediately respond with a fast, immediate, and involuntary reflex response. A complex pathway will include many levels of sensory and motor processing before a response is made. So you have the same stimulus to the same receptor to the same sensory cell, but instead of going through a single central nervous system neuron before the motor activation, it will go up to many areas of the brain. We'll have input, attention gating, recognition, association areas, all of the complexities of sensory processing that lead to the different perceptions that are very individual. Each cell added here increases processing time and complexity of the reaction. So a reflex will be a rapid, predictable motor response to a particular sensory stimulus. These are important clinically to test that both our sensations and our responses are intact. So here, for example, is the knee-jerk reflex where you tap on the patellar tendon, which has a stretch receptor within it. That stretch receptor goes through the dorsal horn of the spinal cord and then immediately synapses on and activates a motor neuron that causes contraction of the quadricep muscle. And that causes you to kick your knee out, and then you can test that that pathway is intact. So this is a general reflex arc where you have a receptor detecting a stimulus, the sensory neuron relaying information about that stimulus to the central nervous system, and then the motor neuron sending information out to respond to that stimulus. We can classify reflex based on whether they go through spinal nerves or cranial nerves. Those will be spinal or cranial reflexes. We can classify them based on whether we're born with them. Those are innate, or whether we acquire them. That's why I put this picture of the dog drooling, right? So he has learned to respond reflexively to the sight of food with his drooling response. That's acquired. How many cells are involved? Is it just 2 cells and 1 synapse? That's monosynaptic. Is it multiple cells and multiple synapses? Those are polysynaptic. And what does it control? Does it control skeletal muscle or does it control organs and glands? Skeletal muscle would be somatic, visceral would be organs and glands. So the withdrawal reflex is a simple reflex response integrated by the spinal cord to prevent injury when in contact with a painful stimulus. So let's say you have a pain receptor in your hand and you press your hand on a thumbtack. That pain receptor will send information through the sensory neuron from the hand to your spinal cord. That will be integrated by a single interneuron in the spinal cord, and you will contract your biceps brachii muscle to remove your hand from that painful stimulus. We can also have that same sensory stimulus, which is that thumbtack piercing the skin, activating the same receptor, the pain receptor on the skin of your hand, traveling through the spinal cord dorsal root, just like it did for the withdrawal reflex, but instead we can then send it up to the brain at the same time. It'll cross over the spinal cord, go to the lateral white columns, travel up the brain through the lateral spinothalamic tract that I showed you guys a few slides ago. Synapse in the thalamus, travel to the primary somatosensory cortex, where you will actually become aware of the stimulus. So in the reflex, you withdraw your hand without being aware of it. Then you take a second and think, oh man, that hurt, right? That awareness of the pain in your hand is a separate sensory complex pathway. What we'll talk about next time is how we then produce a motor response and how that gets processed in the brain. So we'll take the awareness of the pain in the hand, that, oh man, that really hurt, to the primary somatosensory cortex, to the motor cortex, to some language areas, from Broca's area through the cranial nerves out to the muscles of the tongue and face, to contract the jaw, facial, tongue muscles, and then you would say, ouch. So each of these pathways involved various areas of the brain, spinal cord, and various receptors in the peripheral nervous system to be able to sense the stimuli and to respond to them. Okay, that was a long lecture. I hope you guys got through it all. Please let me know if you have any questions.